and today I'm going to talk about molecular technique, which is Western blotting and immunohistochemistry. To start with, Western blotting and immunohistochemistry is all about seeing proteins. It's about proteins, and we call it protein work. Immunohistochemistry is done with, is performed mostly with tissues, but Western blotting is done with various types of samples, including tissues, cell lysates, and everything. So it's pretty much done every day in the lab. So you should understand what Western blotting is, understand the general process and the object of doing it. Immunohistochemistry, some labs do it in their own lab, but in our case, there is Immunohistochemistry Center usually, so we give them a sample to ask for immunohistochemistry. However, in doing that, we have to understand what immunohistochemistry is, because we have to ask in details, and sometimes they give you paraffin block, sometimes they give you slide with tissue on. So if you want to work with immunohistochemistry, even with the service center, you have to understand the general process and the object of it. To start with, I'd like to briefly go over the definition of Western blot. Western blot is the process to detect the specific proteins in a sample of tissue or cell lysates, which is cultured in vitro or other things. In general, in most cases, you make a sample with the cell after inducing lysis on the whole cell. So in the cells, in the sample, there's gonna be all the proteins from the whole cell. But for example, if you want to localize the cell, or if you want to see the proteins existing only in nucleus, you can make a sample after nucleus extraction and inducing a lysis on nucleus. Then you will get a protein sample of the nucleus only. So there can be various types of samples. Well, in terms of IP, immunoprecipitation, you can pull the whole protein pool with specific antibody. If you want to see if protein A and B binds together, you can pull the protein sample with antibody of protein A, and if they bind, you can detect protein B from the sample pulled with protein A, because they are a complex. And the last step is, in that case, the sample for Western blot can be the protein that makes complex with protein A. However, there are various cases, but what's important is in Western blot you can detect specific proteins with the help of antibody, and you can actually compare the amount of protein with Western blot. So I'd like to go over the example of Western blot first. This is from the paper published in 2005 in JCI. Well, I don't know the exact context of this paper, but if you see this figure, they first compare the mRNA expression level of this protein FOXO, this mRNA expression can be compared with PCR. Then they compared the amount of protein of FOXO with Western blot here. You can compare the quantity of protein with Western blot, and also you can detect the phospho form of the protein. You can see that you can see the amount of FOXO1 here. However, you can see also the amount of phospho-FOXO1 here. And sometimes we overexpress those protein, and here in a lot of cases we overexpress some proteins tagged with flag or HA in the cells. In that case, since the protein has a tag, it's a little bit heavier than the endogenous protein made. So that's what it says. We are gonna go over what this site means after we go over the procedure of Western blot. Oh. So moving on to the procedure, Western blot starts with SDS page. It's one kind of gel electrophoresis. The protein sample contains thousands of proteins or more than that existing in the cell or tissue. You make a loading sample with those protein sample, and you load those proteins here in the comb. And then you're gonna go over this SDS page in detail after I explain gel electrophoresis. But here what's important is with the charge on, the protein is gonna move. The protein is gonna enter the gel, and it's gonna be separated according to their size. And in this scheme, we use SDS and polyacrylamide. So this is called SDS, polyacrylamide gel electrophoresis. But moreover, in general, is one kind of gel electrophoresis. And what gel electrophoresis is, this is the sample I talked about. With the protein sample, you make a loading sample. In making loading sample, you try to make this loading sample containing the same amount of protein, because without same number of protein, you can't actually compare the amount of specific protein between these two main. So in loading sample, you have to make it contain the exact the same same amount of protein between samples. And gel electrophoresis is the technique used to separate proteins with the help of gel and with the help of charge. Here, the plus charge goes to the bottom, and the minus charge goes to the top. And when this loading sample, which contains various types of protein, undergoes gel electrophoresis, when with the help of charge, they enter the gel and they get separated according to their size. As you can see here, this is what's about molecular weight, And you can see the marker here. And in the cells, there are numerous proteins with all different sizes. However, after purification, you can see only one band, meaning that only one protein is left. That this protein pool got purified well. And this is gel electrophoresis. And in Western blot, we more specifically, we use SDS and polyacrylamide. So in short, we call it SDS page, which is an abbreviation of SDS, polyacrylamide gel electrophoresis. As you can see, this is a gel cassette, and the anode goes to the plus charge goes to the bottom, because this SDS I'm going to talk about has a minus charge, and this negative charge coated protein is gonna be attracted to positive charge. So with that attraction, with the force, the protein is gonna move to the bottom, and it's gonna be separated according to its molecular size, because heavy molecules are slow. So in detail, it's a gel cassette and sample loaded into gel by pipette like this. And the positive charge goes to the bottom. And what SDS is, you can see this pink negative charge here. This pink charge is what SDS does. Negatively charged SDS molecules. SDS has a negative charge, and with SDS in the sample, SDS protein is coated with SDS, and along with SDS we add mercaptoethanol, and we heat the protein samples. With mercaptoethanol and heat, protein usually has a round shape, and this shape determines affects the speed of moving too. So what we want is to have a linear straight shape, with minus charge pushing each other, and mercaptoethanol, which is a reducing agent, which breaks disulfide bond, and heat, those actions make the proteins linear. So you can see that with the help of SDS, charge per mass becomes same for every protein. And charge per mass is one factor important in deciding the moving speed of the protein. And mercaptoethanol makes the protein shape linear. So protein shape is also another factor affecting protein moving speed. So with the help of mercaptoethanol, protein shape becomes same in every protein in the sample. So when those factors are erased from our protein speed affecting lists, it is only mass that contributes to the speed of protein moving to the positive side. So that's why even though protein has very different shapes and very different charge per mass, Western blot can separate, SDS page can separate those pool of proteins only according to the mass. So with sample loaded here, we studied about the factors that induce the changes in protein moving speed and how we deal with it, especially with SDS and mercaptoethanol. Now let's look into how gel is shaped. Gel is more specifically polyacrylamide gel, meaning that this gel inside the gel there's polyacrylamide, and it performs as a molecular sieve. When there is a protein mixture here, and it's like a molecular sieve, so the small protein can pass the gel faster than the bigger protein. So this polyacrylamide gel can work as a molecular sieve and help separate the protein mixture according to the size. So we talked about the molecular sieve. And if you look into gel, you can see that this gel consists of two parts, which is stacking gel and separating gel. And I'd like to apologize for the typo here. It's actually anode. But what you have to remember is that this part has the positive charge. Well, it actually consists of two parts, stacking gel and the separating gel. Oh, this has a typo here. So this stacking gel is for is to make the proteins lined here. When we load the protein mixture samples here, well, according to our theory, they're gonna run with different speed according to their size, and that's how we separate this protein mixture. But to do that, you have to start them running in the same line. Protein start running from here and protein running from here. Even if they have the same molecular weight, they will end up in different location. In that case, the separation process is not going to work well. So to make this located in line, we use stacking gel. In stacking gel and separating gel, the composition is same. They consist of glycine and protein mixtures and Cl-. However, what's different between stacking gel and separating gel is the pH. You know that amino acid is sweeter ion. Sweeter ion, it means they can be positive, they can be negative according to the pH. So in low pH, glycine becomes more positive because H plus, hydrogen H plus got attached to glycine. However, in high pH, they lose H plus, they become more negative in charge. So you can see the order of glycine, protein, and Cl- changes. Since it has the strongest negative charge, it comes to the most bottom here. So in stacking gel, whose pH is 6.8, glycine is much more negative because due to the fact that amino acid is sweeter ion. It comes here. So the protein is packed between glycine and Cl-. So it's gonna be, it's like a sandwich, and with the charge on proteins, it's going to be squeezed between glycine and Cl minus. So it's going to be, it has this height, but it's going to be squeezed, and after they pass stacking gel, it's going to be lined up like this in linear shape. However, in separating gel, due to the fact that glycine is zeta ion, and in high pH they lose H plus and they become more negative, they come the order of protein, glycine, and Cl minus changes. They order in this order. They line up in this order. So protein is now not squeezed. They get free from this two molecule, so they can move according to their own speed. And due to what I taught, due to the fact that this gel is polyacrylamide gel and it works as a molecular sieve, they move with different speed according to their molecular weight. We can successfully separate those protein mixture according to the size. After SDS page, we perform transfer. We transfer this gel, the proteins separated according to their size in the gel to the membrane. We move the protein in the gel to the membrane because it's hard to deal with gel throughout the whole step. We transfer those proteins in the gel to the membrane. We call it transfer step. We also use charges since proteins are coated with SDS, which has negative charge. If we make this membrane side positive, the protein is gonna be attracted to this side, and protein is gonna be bound to membrane. Here, after the transfer step, we're gonna have membrane, and on that membrane we're gonna have protein on it, and there's gonna be millions of proteins here, and we want to see the specific protein. To see the specific protein, we use antibody. For example, if we want to see GAPDH, which is a glycolysis enzyme, we use primary antibody as the one detecting only GAPDH. And with this step, we're gonna have this protein and primary antibody complex. And then we attach secondary antibody, which detects the end of primary antibody. And in the end of it, there is an enzyme, and it can induce some reaction, and we can see the bend. But before this primary antibody attaching step, we usually go over this blocking step. So transfer, blocking, primary antibody, secondary antibody, and detection step. This blocking step is usually we use skim milk or bovine serum albumin as a blocking agent. This primary antibody, we call it specific, but there is unspecific there can be a unspecific binding to the proteins here. To inhibit this unspecific binding to the protein of the antibody, we coat this protein with BSA or many unspecific proteins in skim milk. In that case, those proteins is rather gonna have unspecific bind to the protein, and the chance that primary antibody is gonna have unspecific binding to the proteins on the membrane decreases a lot. So before we attach primary antibody, we go over blocking step, and then primary antibody, and then secondary antibody, then we detect the signal. Well, let's go over, after I explained the general process of Western blot, let's go over the example of Western blot once again. This is example the result of PCR, which sees mRNA expression level, and BCD is about Western blot. We can also see phosphoform. If we use an antibody which can only detect phosphoform of the protein, we can see the we can compare the amount of phosphorylated form of the protein. Here, they are comparing the level of FOXO1. Since they overexpress FOXO1 tagged with flag, in the third lane, the amount of FOXO1 increases a lot compared to the other lanes. Here, it says tagged and in endogenous protein. Tagged protein is what's tagged with flag or HA. Since it's tagged with something, it's a little bit heavier than the endogenous protein FOXO1. So since Western blot is separating based on the molecular size, their size is gonna be a little bit different. They're a little bit heavier than the endogenous protein, and they're gonna be located a little bit upper side. They're gonna be located in the upper side of the endogenous protein. Actually, flag is not that big. Western blot is that sensitive. And they see phosphorylated form of FOXO1 as along with seeing the level of FOXO1 protein. Well, what's impossible with PCR is that they cannot see the phosphorylated form of something. Western blot is the only thing you can compare the phosphorylated version of the protein, and in many cases proteins are there, but they are activated through phosphorylation. So Western blot is very important in comparing the activation of something. And next we're going to talk about immunohistochemistry. Immunohistochemistry is a process to identify specific proteins in a tissue. It's mostly it's done in tissue. So many clinicians, they have an access to a tissue sample, so they use a lot of immunohistochemistry. Well, this is an example of immunohistochemistry. You can see that here they are trying to talk about this idiopathic pulmonary fibrosis patient. In IPF patient, they use this PDCD5 antibody. They're saying that in IPF patient, in patient lung tissue, PDCD5 level is unregulated. With immunohistochemistry, you can also see the localization of the protein, as you can see here. This is immunofluorescence image. Immunohistochemistry makes colorization of the protein. However, immunofluorescence, it's similar with immunohistochemistry, but it has a fluorescence dye on it, so you can see in this fluorescence image. As a process, well, I got the image from this creativediagnostics.com. As a general process, we cut the tissue like this, we fix with formaldehyde in most cases, but there are many fixation agents. And then we embed this tissue in paraffin, and then we can get this paraffin block. Paraffin block is important because with paraffin block you can section this block later, and then you can stain it with your protein of interest later. So in many cases I preserve this tissue with this paraffin block because my protein of interest changes over time. After paraffin embedding, we section them, we deparaffin them because paraffin interferes with staining process. We go over antigen retrieval step, and like Western blot, we go over blocking step, and then we attach first primary antibody to it and secondary antibody to it, which detects the end of the primary antibody, and we go over the detection step. This is done on the slide you can see here. So immunohistochemistry is a process to identify specific proteins in a tissue. We went over the general process. Sometimes when you apply for the service of immunohistochemistry, they ask you if you want to get your tissue back with paraffin block or the slide. Paraffin block is the we make paraffin block, or you can make cryo block, depending on the staining you want to do. But in most cases, you get paraffin block back with the tissue. You can also get slides. This is basically same with the paraffin block, sectioned and attached to the slide. And then we go over the paraffination step, and as if it's in Western blot, we go over blocking, we attach primary antibody and the secondary antibody, and we detect the signal. Here, and another advantage of immunohistochemistry is that you can see the localization of the protein. Lungs are made of various structures and various tissues. Here they are talking about club cell specific role of PDC-D5, and the start point is this immunohistochemistry. You can see you can not only compare the amount of protein between two conditions, you can see the localization of the protein. And when you detect, we used colorimetric process to have this image, but if you attach fluorescence dye, you can have this fluorescence image. It's much more the image from immunofluorescence can be much more high resolution compared to these colorimetric rays. So it's good way to see the co-localization of two proteins. This is the end of my lecture. I would like to apologize. First, I wanted to show my face and give a lecture to all of you, but currently I'm in quarantine. I'm at home, so I wasn't able to go to my office, so excuse me for giving this lecture with only my voice. I hope that next time we can actually meet and discuss in person. Thank you very much.